So let's turn our attention from the overall structure and anatomy of the eye and focus in on the retina itself. Now, we sometimes think of the retina as just this structure that contains nothing but photosensitive cells. We might even think of it sort of relatively homogeneous. But in fact, it's a complex structure containing a number of different cells. This is divided into 7 individual layers by neuroanatomists. And it contains 2 different classes of photosensitive cells, and within that second class, 3 different kinds of photosensitive cells. I'm not going to go into a tremendous amount of detail on this, but we will look at it. And the first thing we'll want to notice here is this. Light is coming from this direction. This is the front of the eye. This is the front of the retina, and this is the back of the retina. So light comes through the eye, goes through the vitreous humor, and it has to penetrate through all of these different layers here to get to the rod and cone layer, which is the part of the rods and cones, these little discs here, that contain the photosensitive molecules. So it's not an intuitive design. The photosensitive part of the retina is actually pointed towards the back of the eye, and it's covered by a number of different layers of different other cells. So let's briefly look at these, and then much more detail is given in the eye lab if you choose to do that. So we have the nerve fiber layer, and this is where the axons, the invaginations from the ganglion cells, go out and go together to form the optic nerve leaving the eye. The ganglion cell layer contains the ganglion cell bodies, and then they synapse on connect to cells in 3 different layers: the inner plexiform layer, the inner nuclear membrane, and the outer plexiform layer. And these 3 layers contain a number of different cells. And it's important to realize that all of these cells are processing the information about light intensity that gets transduced by the rods and cones back here. So this isn't just a photosensitive structure. Right? This is also an information processing structure. It's not just transducing or sensing the light that's being projected into your eye from the visual scene. Right? It is also processing that, sort of first run processing. And there are a number of different cells that are operant in here as part of that processing. Once we get to the outer nuclear membrane, the outer limiting membrane, those are the bodies of the rods and cones. The rod and cone layer contains the actual photosensitive parts of the rods and cones, those little discs that contain the photosensitive molecule that gets changed, reconfigured by the absorption of 1 or more photons. In rods, that is called rhodopsin, and there are different opsin versions for different cones. And then finally, we have this pigment layer at the back of the eye that absorbs light, prevents it from flowing back and rather bouncing back and activating rods and cones. So the most photosensitive part of your retina there is pointed towards the back of the retina, away from the light. And part of that's for metabolic reasons. Part of that is to prevent a bunch of noise from light bouncing around and so on. Right? So this pigment layer is dark for us. And of course, our rods and cones then, they are the things that get activated by photons. We have 2 major classes. We have the rods, which are larger and much more numerous in the retina. They have a greater sensitivity. A rod can be activated by as little as a single photon under the right conditions. And then we have the cones, those 3 kinds of cells that are smaller, that are less sensitive to light. It takes a couple of photons at least to activate them, and that have a different selective sensitivity. They're tuned more to a specific wavelength. So here is our black rod and cone wavelength, right? Black rod wavelength, going from about 400 to about 625 nanometers. Here's our blue or short wavelength cone sensitivity, our medium or green, and our red or long. And they each have areas where they're maximally sensitive. that is, wavelengths that they're particularly sensitive to. But they have a wider range of activation, so that light of other wavelengths will activate them, though this one activates them the most. And they have a significant overlap with one another. So that's a little abstract diagram depicting the retina. This is nice because it's a picture here from Rachel Wong's lab at the University of Washington. And this is an actual picture of a mouse retina. And so it gives you a sense of what the real biological organism looks like, right? So again, here is the front of the eye, here is the back of the eye, and this is our retina. This is the ganglion cell layer, right? Here we have those intermediate layers with cells that process the information, and here we have the rods and cones collected together. And it's important to sort of notice just how messy and haphazard this is compared to those kind of sanitized and idealized diagrams that we tend to look at. So let's focus now on the light sensitivity components of the retina, right? Those rods and cones. Each of them, as I noted, has a selective sensitivity curve. So our short wavelength cone here, also called the blue cone, it has a selective sensitivity curve. It's maximally sensitive to light that is 437 nanometers in wavelength. Our rods have a very wide sensitivity curve, but they're maximally sensitive to light at 498 nanometers. Green, 533, and red, 564 nanometers. Now they will fire for light, just more light is required, or they'll fire less intensely because they have a graded response to light anywhere along these curves, anywhere within their selective sensitivity curve. They fire most strongly with the least stimulation for the wavelength that they have at the top or the peak of their sensitivity curve. And these sensitivity curves overlap significantly. Rods, which are the most sensitive, are also the most common cell in your retina. The retina isn't homogeneous in this way, even a little bit. Right? 120 million of those cells are going to be rods. Only about 6 million are going to be cones. Of those, most of them will be green and red, right? Medium and long range cones. Those are the only cones that are in the fovea. The blue cones are on the outside of the fovea and other parts of the retina. So we have these idealized absorption curves for each cell, and they're drawn against the spectrum of visible light here, which runs from about 360 maybe to about 60, 70, 80. So this is the spectrum of the this is the part of the electromagnetic spectrum that our rods and cones are actually sensitive to. And as I noted, rods differ both in structure and photopigment. The light-sensitive molecules from cones that each individual rod and cone has as its primary photopigment. Now, again, this isn't a homogeneous structure of the retina. Cones are neither present in equal numbers nor distributed evenly in the eye. Long wavelength cones, these red cones, constitute 64% of all the cones in the average human eye. 32% are classified as these medium range cones, and only 4% are classified as the short wavelength cone or blue cone. All of these cones, all of these photoreceptors hyperpolarize to light, and they exhibit a graded response depending on the wavelength and the amount of light that is getting to them. I should also mention that there are no blue cones, those short wavelength cones, in the fovea. They're in the periphery, but not in the fovea itself.